Good morning, everyone. The webcast will start now. This webcast I'll be giving is at the title Pressure Ulcers Research from Seated Posture to Buttock Tissue Strain. My name is Christian, and I'm from the Anybody Research Group at the Department of Mechanical and Manufacturing Engineering at Aalborg University here in Denmark. This work is part of my PhD project, which I've been doing for the last 2.5 years, and that kind of means that I have 6 months left, so I'm kind of in a hurry. Before we start, I would like you to know that you can all ask questions during this presentation, and you can do that by launching the Q&A panel down from the menu, and then you can type in your questions, and then you can see the answers next to your questions. And make sure that before you send your question that you ask the host, presenter, and panelist, because then everyone here gets your question, and someone will be able to answer it. If it's a quick answer question, then you will get the answer right away. And if it's a more another question that's more broad and related to this research, then I'll try and answer it afterwards. So please ask questions during this presentation. On the right side, that's a picture of me, and I'm the presenter of this webcast. Where on your left side you have Anne Kies, who's the host of this webcast, and is another picture of Søren Tørholm, and he's the panelist, and he'll be answering questions as we go on. First, I'd like to start with a few of the hypotheses of why these pressure ulcers, why they develop under a buttock, for example, when you sit in, for example, a wheelchair or lie in a bed. And basically, there have been two hypotheses that have kind of ruled the research area. And the first of them is the ischemia, which is basically that you when you sit on for example, your buttocks, then the tissue is compressed, and that means that the arteries are also compressed, and then there's no blood flow through these arteries, and that means that the tissue will lack oxygen, nutrients, and waste products will also not be removed from the tissue. And this idea has been the largest player within these two hypotheses for the last many years. The other hypothesis that is kind of is more and more accepted, and especially in the research areas within pressure ulcers, is the deformation. And this idea is just that the cells die due to pure deformation. So when you sit on the buttocks, it's deformed, and then some of these deformations the cells cannot tolerate, and then they basically die. So I'd like to dig a little bit deeper into these two hypotheses, and then I'd like to highlight these two publications. The top one is from 2006, and that study was done by Casimir FE-model and from this Dutch research group, and they looked into the relative contribution of compression and hypoxia when to develop these muscle tissue damage. And basically, this means that they had cells growing in the lab, and then some of them they compressed, and some of them they didn't allow to have any oxygen, and then they saw which one of them died the first, basically. and they found that deformation is the most important factor, and it's much more important than the oxygen. They could starve the cells for up to 22 hours before something happened with these cells, and it didn't really add a lot of effect if they both deformed them and also subjected them to hypoxia. The bottom one is of these two papers is from an Israeli group, and they have done computer models and trying to simulate what happens to a cell when you compress it. And basically cells are like water balloons. They are incompressible because most of them consist of water. And as you probably all know, if you squeeze a water balloon, it will extend in the other direction. And they basically saw the same thing in this study. And then if you keep on squeezing a water balloon, at some point the membrane or the balloon will burst and you will get wet. And it's essentially the same thing that happens in this what they show in this study. The idea is that when you extend this membrane, then you open some of these. Basically, there's a lot of holes where you can transfer different things over the cell membrane, and basically some of these gaps or ports are becoming bigger maybe, and some of the stuff that's not supposed to be inside the cell comes in, and something goes out that's not supposed to go out. No one is really sure of what is going in and out, and that is not supposed to do that. But this research group led by Amit Geffen is currently doing studies on this area. Now I'd like to lead your attention to a paper we wrote that was published in the spring. It's called Missing Links in Pressure Ulcer Research and Interdisciplinary Overview. So basically that's about a lot about this talk essentially and this webcast, and you will find a lot of the references and so on in this one. So moving on a little bit, this presentation will kind of look like this. I showed here the picture, and we'll start from over here, where we have a computer model sitting in a chair, and that can calculate reaction forces between the seat and the body. And those reaction forces are then transferred into to this model over here, which is another type of model. I'll go into these areas. That's kind of the whole webcast, and that this model can then cap the tissue strain, which you can see an example of down here, and the tissue strain can then be related to some kind of a cell death. So, for example, saying that the cells cannot tolerate a specific type of strain or a specific amount. And then you can compare so what the cells can tolerate with what we calculate from this given seated posture. So this is basically what the presentation will be about. So starting from the left corner up there, we have this seated human model, which is an antibody model. So it's made in the antibody modeling system. And as you see here on the right here, it's sitting in a chair. It's a generic person sitting in a generic chair that can be changed in whatever way you would like. So you can change the seat angle or the backrest angle and the height of the footrest, all the stuff that you would do in a normal chair or a normal wheelchair or something like that. The model was developed a few years ago, and actually at this web page where you also found this webcast, there's another webcast done by Professor John Rasmussen, who did a webcast on the development of this model. And actually, there's another one also by which I held, I think, around a year ago on the validation of this model. I'll show brief validation results in this webcast as well. Yeah, so basically we have this seated model over here that you can change into whatever posture you would like and sit in whatever chair you would basically like. You can change friction coefficients of the support surfaces and so on as well. So the validation, I said I only have this one slide, so if you already saw this work, then bear with me for a second or two. So basically, we did a lot of experiments with three different subjects sitting in a lot of different postures. And as you can see over here on the picture, we have this wheelchair, and it's actually mounted with force measuring equipment. So we have a force plate in the floor and a force plate as the seat, and also force measuring equipment in the backrest here. So we could measure all these forces. And then, as you can see, the subject has all these white dots on, and that's for the motion capture system. So we can record the posture. And this posture that the person was sitting in was then transferred into a anybody model, as you can see right next to it. So this model is sitting in the same way as the subject was doing in the experiment. And then basically you can see these blue lines that goes out from the anybody model, which represent the forces, the reaction forces between the seat and the person. And those forces could then be compared with the ones that we could measure in the experiment. And a comparison could look like this, where we have the orange one, the yellow ones are the model, and the blue ones are the experiment. And up the y-axis we have the shear force on the seat. So that's the parallel force on this seat. And the x-axis would change the seat angle. So this is just changing one parameter, and then you can measure it at the other one. And as you can see, they correspond very well. So as I said, you could take this model, and then you can change the seated posture and the angles between the backrest and also the seat and so on. And then you could calculate the reaction forces between the chair and the body. And as this is a bit closer view, and you can see that you have a normal force and a shear force for each of these. The blue lines represent the forces. So just a little example for what you can how you can see these things changes. I've done this parameter study where we have the seat angle on the x-axis and we have the backrest angle on the y-axis. And then the nice colors, as you see, is the shear force on the seat. And the shear forces on the seat there's a reason to be so concerned about that one. This is considered one of the risk factors for developing a pressure ulcer when you sit in a chair. So really, you would like to be around this area where you have zero shear force, meaning that up through this red line is kind of the place to be. And in order to illustrate it a little better, I made a couple of models showing that over here in this corner we have this model where you have the backrest is upright and it's leaned very much back in the seat. Over here in the other corner you have the seat is tilted forward and the backrest is still vertical. Up here in the far left corner you have the backrest and the seat tilted backwards. And over here in the last one, it's the backrest tilted backwards and the seat is tilted forward. So basically, then you can go in and say, well, I have a seat angle of 15 degrees. So I would like to go up here. That means I have to go in here, and my backrest angle can be 33 or 34 degrees. Um, so this is just for one subject and for one person, and it's just for the normal anybody model. So that's something like a 50 percentile European male. And things could change a little bit if you change the properties of the model. Um, so again, I would just like to mention to you guys that you can actually ask questions during this presentation. And you do that by launching the Q&A panel from the menu bar, and then you can type in your question. But before you type in before you send it, you have to make sure that it's sent to host, presenter, and panelist in order to make everyone here see the question so they can answer it. And some of the questions I will be answering afterwards after this presentation, where you're also able to ask questions again. Yeah. So moving a bit on, coming into the next part of the presentation, which is the finite element model of the buttocks. So this model is developed by and owned by a Würfel engineering company in South Germany. It's called the Casimir automotive model. So from that name, you can kind of guess that it's mostly used for automotive seating. It's a non-linear model, so it has kind of advanced material properties. And as I said, it's used mostly for automotive seating, and especially vibration analysis is the main topic there. So basically, it's a continuum model with including bones, soft tissue, and some muscles for posture as well. And basically, you could say that the strain of the tissue is very depending on the position on that you put the subject into in the chair. So in an automotive seat, that is that's a lot of you say the gravity is very important because you lean quite a lot back. So that's not really a problem there. But if you sit in a normal chair or wheelchair, then there might be other things that's not as easy. And that's why we try to we make a collaboration with Würfel in Germany to make this project basically. So the interface between this the seated anybody model and the Casimir automotive model, I tried to list up a few things here that are very nice for the two models. You could say the anybody model is dependent on the posture basically, and it can then calculate the reaction forces that's acting between for example, the seat and the subject sitting on the seat. It's also very fast calculations that easily can be done on a normal laptop. The Casimir model, on the other hand, has very detailed tissue strain and very detailed tissue modeling, and it's also dependent on boundary conditions, you could say, where the antibody model doesn't have any soft tissue. It's a totally rigid body model, and however the Casimir model does. The thing that you could say the Casimir model is very dependent on the boundary conditions, and from that I mean that that it's very important where you sit the person in, and you could say that these boundary conditions can be taken from the anybody model because it actually calculates these boundary conditions. So basically, the boundary conditions can be calculated by the antibody model and feed it into the Casimir model, and then this detailed tissue strain is basically something that can go the other way, because the antibody model doesn't have any soft tissue. So just a very preliminary example. It's work in progress. As you see, the mesh is not so fine, because that's not something that has been important for the stuff the Casimir model has been used to so far. However, in this study, we need to change that a bit. But this example is just showing that on your left side there is the model is subjected to gravity only, and on your right side there's added a shear force on the seat, or you could say on the backrest. And what you can see is you have the shear strain in the tissue. It goes from, as you see over here, it goes from red to zeros around orange, and then it becomes green and blue, and which is worse and worse. And you can see that basically this area is a bit more green and yellow than this one. However, it is a very preliminary, and we would like to show some results when we are further in the progress, because it is work in progress. And you could say, to sum it up, this model, this final element model, needs kind of three things to work, and to make a good final element model. You need a good geometry, and there we use the geometry of the Casimir model. Then you need material properties, and that is also handled by the Casimir model. And then you need the boundary conditions, so the way you load the model. And then that part is then handled by the antibody model. So basically, then we need to refine the mesh and to calculate the tissue strains. And in that way, we can relate the posture from the antibody model with the tissue strain from the Casimir model. And why is that important then? Well, tissue strain is important because, as started, I mentioned in the beginning, I mentioned that cells die due to deformation. So the thing is, what type of deformation? Because the thing is that tissue is nearly incompressible. That's why I gave the analog to the water balloon, where you squeeze it and then it extends in the other direction. So I just have a little example here where you have the yellow box. You can see if you compress that, then it becomes wider in the other direction, because the volume is basically constant, or the area in this case is constant. If you pull it and if you apply attention to it, then it essentially becomes the same shape, just rotated 90 degrees. And then there's another thing you could do to it. You can apply shear, or shear strain, and that gives a shape like that one over here. The thing is that shear strain is also, when you compress it, as you do in the middle picture here, that's also shear strain. And I can show that by adding a red square that's just rotated 45 degrees, and then we try to compress it, and it then will look like this. And you might see the relations between that shape and this shape. They're actually very similar. So this just shows that the shear strain is also there when you just compress it from one side, so uniaxial. Yeah. Another thing is that you can add a hydrostatic pressure to a water balloon or a body, and if you do that, that basically means that you apply pressure from all sides, and it's the same pressure. So, for example, diving into to the swimming pool, that will apply hydrostatic pressure to the whole body. And that's kind of another thing, because then you don't have the only thing you don't have here is actually shear. So there's no shear strain when you apply that hydrostatic pressure. So a couple of studies that I would like to show that's been working in this area of mechanical test of cells. They are mentioned down here, and basically, if you look on the right picture here, then you have some tissue growing down here, and then you have an indenter that you press into this tissue, and then you can look at it from underneath, and then you can, for example, get plots as you have over here to your left side, where the dark pictures represent cells that are living basically, and the white dots that show up after some hours at least are then the dead cells. So if you look carefully on this on these pictures, then there's actually kind of there's a line around like a round line around these photos, and especially down here you really see the clear bright line of dead cells, and basically that's on the point of that's on the edge of this indenter. So at that point you actually have a large shear strain. Yeah. So basically these studies, they in a way they show that shear strain is something that is maybe considered a key point in in the understanding of what cells can tolerate. And other studies have actually showed kind of similar results, where this is a kind of a early study. It was done in 1953, I think. This guy Hussein, and he did some very nice experiments on rats and guinea pigs, where he applied pressure in different ways, and one way could be a point load on the tissue, and another way could be on the hydrostatic load. The point load has shear strain in it, and the hydrostatic one has not. And what he found was there was a very, very large difference on what the tissue could tolerate regards to the point load compared to the hydrostatic one. So basically this work with these cell experiments, we're trying to. I don't have any results from our own work, but we're also trying to work in this area and create some kind of a cell death criteria that could relate to the tissue strain. So basically, again, I would like to sum up this presentation, and to begin with, we saw how the seated posture could change the reaction forces, and that basically just meant that when you lean back your backrest or change the seat angle, then you basically change the forces between the chair and the body. Those could be transferred into to a model of the soft tissue, for example, this Casimir model, that can then calculate the tissue strain, as you see down here. And the tissue strain could then be related to what the cells can tolerate with respect to the strain, and that could be done by these. It could maybe be showed by these experiments, and then we can compare that to the results from the final element model. And if we add one more thing, then we can actually start to change the posture in order to change the tissue strain compared with the cell death criteria. So we could optimize the posture, so the the tissue strain um, is not as what you could say dangerous for the cells as and in that way we could optimize the posture in order to not um, you could say in order to minimize the risk of getting a pressure ulcer. So in the end, I would like to acknowledge all my co-workers and basically our sponsors of this project.